Good day. My name is Paul Wax. I'm the Executive Director of the American College of Medical Toxicology, and I'd like to welcome you all to our webinar series entitled Medical and Public Health Considerations of COVID-19. Today's webinar is on the therapeutics and their toxicities associated with treatments for COVID-19. I'd like to thank our webinar series partners for helping to promote this webinar series. Much appreciated. Next slide. This webinar is being recorded and will be posted to the ACMT webinar page by Friday. All the registrants should get an email once it's posted with the webinar link. Many of these webinars, we're also developing a Q&A, which can be also accessed on the ACMT COVID-19 response page. And finally, if you have any questions about the webinar series, feel free to communicate with us at webinars@acmt.net. Next slide. There will be a question and answer session at the end of the presentation. This is often a very robust component to our proceedings, and we welcome you to submit your questions. They can be put into the Q&A box or the chat box. And for those of you on YouTube, you can also put them into the chat box on YouTube. Next slide. Our speaker today does not have any conflicts of interest to disclose. Next slide. I'd like to thank my co-moderator, Dr. Ziad Kazzi. Dr. Kazzi is a board member of the American College of Medical Toxicology and president of the Middle East and North Africa Clinical Toxicology Association and associate professor at Emory University. Next slide. I'd like to welcome our plenary speaker today. Dr. Michael Chary is an instructor in emergency medicine at the Weill Cornell Medical School. He completed his medical toxicology fellowship training at the Harvard Medical School program in Boston, and has spoken widely about this very important topic of both therapeutics and toxicities associated with COVID-19 treatments. Today he'll be discussing the most evidence-based therapeutics and review reasons why some of the early promising candidates, in fact, have subsequently faded out. Michael?
Thank you very much, Paul, and thanks to Ziad and everybody else at ACMT. And just a quick check that everybody can hear me and can see the slides advancing.
We can hear you fine.
Yeah, all right. And the slides just advanced. So as Dr. Wax mentioned, we're gonna spend some time today talking about some therapeutics, and I'm using the term very broadly, that were used to treat COVID-19. And I'm Mike Chary. I did residency in emergency medicine down at New York, went up to Boston for fellowship and back down to New York again, where I'm tox research and EM faculty at Weill Cornell. So let's begin with the bottom line up front. Of all things, social distancing works to control the spread or to limit the spread of coronavirus or of COVID-19. If that's not possible, and for many of us who are working clinically, you can't stay 6 feet away from everybody, you need appropriate personal protective equipment and frequent hand washing. And that personal protective equipment may include masks and face shields in addition to masks, depending on if you're exposed, for example, to respiratory secretions. Once we get into therapeutics, dexamethasone may prevent intubated patients from deteriorating further, but really nothing else to date changes 28-day mortality, or is better at 2 weeks than just, take 2 ibuprofen and call me in the morning. But absence of proof is not proof of absence. Many of the studies are rushed and confounded, and rushed by nature of the pandemic. We want to, at the first chance we can get an effective therapeutic out. But sometimes people have been enrolled in multiple trials at the same time, or what is called a control arm is they're actually also receiving an antiviral. So it's tough to draw definitive conclusions from this. And when all is said and done, we really need an effective vaccine. And by the way, I will say that BLUF is a term that I learned from my co-fellow, who was a captain in the Army, and he tells me it stands for bottom line up front. Another way of summarizing everything, and this is from the COVID-19 treatment guidelines, which is something that Dr. Fauci has put together. It's an excellent resource. And in particular, I've taken a figure from the treatment recommendation page. And this is what the current recommendations are from the top. You notice that it goes by disease severity, going down by hospitalized and not hospitalized, hospitalized and requires supplemental oxygen, but not on ventilation, hospitalized requires delivery through a high flow device, or is on ECMO. And you'll see two names over and over, dexamethasone and remdesivir. But we started out with more than dexamethasone and remdesivir. And in this slide, to give you an example of some of the ones that were, it seems like ages ago, but in March and April were talked about and just kind of dropped out of the field, I've put this summary slide together. On the columns are the stages of investigation. So in vitro, animals, some preclinical, clinical trials, all the way to a randomized control trial. And then the rows list some selected therapeutics. The therapeutics that have a parentheses T after it we'll be discussing later in a presidential case study. The black color, the black shading, is meant to suggest therapeutics that had already gone through all this preclinical and clinical evaluation, but for another indication and were being repurposed. So dexamethasone or glucocorticoids widely have been used for many purposes and were around for quite a long time before the current epidemic came around. Similarly with azithromycin and hydroxychloroquine. So we started out with a lot of drugs. Some were new. Some were like convalescent plasma is an old idea that's made new again. And many were repurposed. And you can see that in the bottom, I've put three here, Lopinavir, Ritonavir, APN01, which is actually a recombinant humanized ACE antibody against the ACE receptor, and leronlimab, which is an IgG4 used still in some HIV treatment. And all those really showed promising work in vitro, inhibited viral replication, but just didn't progress any further, and there was no translation from those from the animal studies into any clinical efficacy against COVID. So I want to spend a few slides talking about the biology of SARS-CoV-2, the virus that causes COVID. And I want to do this to bring out 2 points. The first is to give everybody a general understanding of where the pressure points are in the life cycle of SARS-CoV-2. And the second is so that we can see when people make these regimens combining one or more therapeutics, we can get a better sense of which combinations make sense. We are familiar with, for example, HIV, the utility of combining multiple therapeutics to, as it were, hit the virus at many stages of its life cycle. And the main reason to do this is to prevent some sort of mutational escape. This is a cartoon from The Economist describing the structure of the coronavirus. And there's a few features of interest here. This slide may be familiar to many in the audience. We see the red spikes, which is a protein on the outer surface, and those red spikes are what gives coronavirus its name. They're also the thing to which almost all of the vaccine-related technologies are directed against. The spike protein is the part of the cell, or is the part of the virus that the virus uses to attach to respiratory cells. So what happens is the spike protein docks into the ACE2 receptor. Now the ACE2 receptor is a protein expressed on many cells, including not only the liver, but the respiratory epithelium. Spike protein docks into this ACE protein, and then this entire virus gets kind of engulfed or endocytosed by the cell. The spike protein has been challenging to raise antibodies against because it's coated in what can essentially be described as a slime of sugar. And this goo is important clinically in a few reasons. The first is that heavily glycosylated proteins tend to provoke just a profound nonspecific immune response, which kind of sounds like the cytokine storm. The second is that despite that a robust nonspecific immune response, they're tough, they're not that immunogenic, which explains why there's a lot of different approaches to raising antibodies against it. From that same article, here's a deeper schematic showing once this coronavirus is docked into the cell, we can see, looking with the number 1 on the upper left, that it uses the ACE2 receptor with the TMPRSS2 co-receptor to get pulled into the cell and be endocytosed. Then in this endosome, it relies on the natural tendency of endosomes to acidify and uses that as a signal to kind of release its contents into the cytoplasm, where the viral RNA hijacks the cellular machinery similar to other viruses. And the take home from this slide is not to make us all virologists, but to see that there's many points along here, and you want to, it stands to reason that you want to choose a few therapeutics that intervene at a couple of points along this until we have a fully effective vaccine. Here is a similar schematic, and what I've done here is overlay where the therapeutics interact. Now, unfortunately, the slides are not perfectly one-to-one, so things are moved around a little bit, but we can see that we have a TMPRSS2 inhibitor, this camostat, which they're still working on some animal trials in. We have the somewhat infamous chloroquine and hydroxychloroquine, which aim to ion trap the virus inside the endosome. So it's inside the cell, but it's inside the cell in a little pouch, and it can't get out of that pouch. For some of the toxicologists on the call, this actually kind of reminded me a lot of kidney toxicity from vancomycin, where the drug keeps getting pushed into the endosome. But we know that vancomycin eventually is nephrotoxic because it kind of leaks out of the endosome, or the endosomes pop. And there's no data on this, but we would kind of expect something like that to limit the efficacy of chloroquine or hydroxychloroquine. Going further downstream, we see the Lopinavir, and there's also what is sometimes used a little more widely in other countries like Japan or the Middle East, the Favipiravir. One stage further, the Lopinavir, Darunavir, these are protease inhibitors, and they do exactly what you'd expect, which is they inhibit protease, and this is a viral protease that's used to break the big polypeptide that comes out of the virus into the small non-structural proteins. Non-structural proteins can be a little bit of a cryptic name. What it means is the matrix protein and the all the things that actually encapsulate the virus, all the components of the virus that aren't the genome, that aren't the spike protein. And I see that the slide is very, very blurred, so I'm sorry for that, and thanks for noting that. Hopefully other slides will be brought into focus a little more. It was a high resolution picture. The general point, which I think persists a little bit without a diagram, although of course would be helpful, is chloroquine acts one thing, downstream Lopinavir acts, downstream of that the Remdesivir and Favipiravir work, and these are Remdesivir, as we'll talk a little bit later, is not dissimilar to acyclovir in the sense of it inhibits the elongation of a nucleotide chain by acting as something that you can't polymerize from. In the case of Remdesivir, it acts as an adenosine analog. And then you have a bunch of IL-6 things, and they're kind of all the way up in the center here. And the idea is that IL-6 signaling, although it's actually needed to make, IL-6 signaling is required to have B cells actually secrete antibodies, which sounds like a good thing. Aberrant IL-6 signaling may be driving the cytokine storm. We'll move onward, and yeah, as Ziad notes, we will get a we'll get a higher quality version in the PDF. So that's the overview of the life cycle. The virus comes in, it has to dock, it's encased in something, it has to get out of the casing, it has to hijack the machinery, and it has to get out of the cell again. And at each point we can do something. I'm going to talk first about non-specific therapies. And when I say non-specific therapies, I mean things that are not specific to the genome necessarily of SARS-CoV-2. So the few ones, and this is not an exhaustive list, but I think it's an illustrative list. The ones we're going to talk about are chloroquine. And when I say chloroquine, I mean also hydroxychloroquine. We're also going to talk about dexamethasone and convalescent plasma. And as we get into the details of each drug, I will say that the information we're going to present is current as of 2 days ago. So the rationale, as I hinted at before, for hydroxychloroquine is that the virus gets into the cell, but you prevent it from getting out of its protective casing, if you will. You do some ion trapping that prevents the lysosome from acidifying, thus removing the signal for the virus to actually release its contents. There was some promising preclinical data, but there was no consistent efficacy in humans. And this hydroxychloroquine was studied very broadly, whether for severe illness, kind of requiring mechanical ventilation, for moderate illness, for post-exposure prophylaxis from somebody who you found out you were next to somebody or possibly breathed on them or were breathed on by them and they were later found out to have COVID. It showed absolutely no change in either severity of illness or 28-day mortality. And I don't want to spend too much time on the side effects of it because I think it's been discussed both nicely in this seminar and in a few papers. Briefly, it can lead to cardiovascular collapse and kind of other things that resemble synchronism, which is not that surprising considering it's a quinine derivative. The toxicity is described to start at about 20 milligrams a kilogram. And the dosing regimens that were used all wound up being for an adult male, your mythical 70 kilogram male, wound up being about 25 milligrams a kilogram. So, already starting out in dangerous water. And indeed, lots of these trials were terminated due to QTC prolongation or recurrent syncope. There's a unique antidote. If there's a cardiovascular collapse, you give your usual pressor dose of epinephrine, but also about 2 milligrams a kilogram. And I would involve your local toxicologist, as it gets pretty esoteric. So moving on to dexamethasone. Dexamethasone is one of the two things for which the strongest evidence exists for its efficacy. The rationale here is that when SARS-CoV-2 docks to the ACE2 receptor, the ACE2 receptor is there for other reasons. It usually is exerting this, what you might consider tonic inhibition against self-destructive lung inflammation. And when the ACE2 receptor is tied up, docking SARS-CoV-2, that inhibition against inflammation is lost. And it was thought or hypothesized that glucocorticoids were one way to rescue that inhibition. and some preclinical trials suggested that of your usual panoply, dexamethasone is the one that rescued ACE2 inhibition the best. To date, the data only show a reduction of mortality in intubated patients and nothing else. And you can, it's not been conclusively demonstrated that that reduction of mortality is due to the rationale above, but there is a reduction of mortality of at least 30%, depending on the study. And I think we're all, or many of us may be familiar with some of the side effects of dexamethasone or glucocorticoids at large, both the neuropsychiatric as well as the endocrine disruption and the hyperglycemia and the kind of Cushing-like syndrome, and with prolonged use, certainly there's decrease in bone density and just a decrease in the thickness of skin. So we'll move to the third one. And convalescent plasma is somewhere between a specific and a non-specific therapy. The rationale, to put it bluntly, is that if somebody made effective antibodies, just kind of use their antibodies. The issue with this is really kind of one of heterogeneity. And just as each infusion of fresh frozen plasma is different, each of these infusions of convalescent plasma depend a lot on the donor, on the viral load of the donor, and on just the general biochemical makeup. To date, no studies show any change in 28-day mortality, although there were some very promising case studies. And the side effects are what you might expect of some transfusion reactions, off-target effects. I will say with the kind of standard modern techniques in blood banking, we don't necessarily, we're not that concerned with the transmission of plasma-borne pathogens, but we still should be vigilant about it. So those are the three non-targeted therapies, hydroxy, that I brought up, hydroxychloroquine, dexamethasone, and convalescent plasma, of which if you're intubated, dexamethasone helps, and none of the three are known to help or are shown to help for any other patient population. So let's move on to targeted therapies. And here's another slide that I hope is not too blurry. But the idea here is to, again, just keep orienting ourselves to the many steps that we can intervene on. And targeted things are going to leverage or make use of the vaccine structure or of the viral genome. And they can be as simple as neutralizing antibodies that are in the upper corner there that just bind to the spike protein and essentially opsonize the virion so that it doesn't ever get into any cell. In combination with that, we have things that are kind of standard armamentarium in virology, protease inhibitors like Lopinavir, and then what are essentially transcriptase inhibitors like Remdesivir or Favipiravir. And then there's an interesting thread, and I'm thankful for the time to be able to talk about this a little more in a few slides. There's kind of a parallel thread that exists mostly in literature coming out of China, where they talk about the impressive immunomodulatory effects of traditional Chinese medicine. And it's something that has not progressed far at all, but I think is worth, as toxicologists and people who need to be aware of the concern and the practices that people might have around treating COVID. I think it's something that's helps to be aware of. So the other one that's on the coronavirus task force recommendations, Remdesivir. The rationale is, as I've suggested before, somewhat by analogy with acyclovir, although we're talking about RNA here, not DNA, is that remdesivir with 3 phosphates looks like ATP. It gets incorporated into the viral RNA by RNA-dependent RNA polymerase, and that leads to premature stopping of the translation. So, the virus has already gotten into the cell, it's gotten past the hydroxychloroquine step, it's gotten past the NRTI step, the protease inhibitor, but you can still, once you get to the NRTI step, you will not make too many functional proteins. And so the idea is that the virions may be released, but they're not necessarily going to be able to infect another cell. And with that attenuation of the infectivity, the host might be able to fight it off without significant morbidity or mortality. So there's still no decrease in severity or 28-day mortality, but there's a bit of an asterisk with this. If you take 200 milligrams by mouth on the first day, and for 4 subsequent days, so a 5-day course, you take remdesivir, you do have less severe symptoms. And this is, most studies have used a 6-point grading system of severity of symptoms, and less severe means goes from like a 5 to a 4 or something, which usually corresponds to going from ICU to step down to floor to discharge. However, the 10-day regimen, which would be 200 milligrams per mouth on the first day and 9 subsequent days, was no better than control. And that's a little interesting because of, I'll respond to that in a second. That's a little interesting because you would expect the 10 day one to be at least comparable to the 5-1, not worse. The therapeutic monitoring, and I'm seeing some of the questions here. Yes, it's been, remdesivir has been used for other things for a while, and we know that you need to look for an elevation in transaminases. There were some very rare reports of acute kidney injury, but it usually has a reversible elevation in your AST and ALT that goes away when you stop it, with, to my knowledge, no permanent histologic changes. There are, off the top of my head, I will confess I don't remember the toxic serum concentrations for remdesivir, but this 200 milligrams in the first day and 100 for each subsequent is quite a common regimen. It's not like the chloroquine one where you're using between 3 and 9 times the usual amount. The main concern with remdesivir is in the elevation in transaminases. Ritonavir and Lopinavir was kind of more talked about, or there was a lot of interest in it early on, and it kind of dropped off the map. The rationale behind this is it's a protease inhibitor, and it works pretty well at inhibiting a protease in SARS-CoV-1 and in MERS. So maybe since that protease has a very similar sequence to the one in SARS-CoV-2, maybe ritonavir would do something with that. Lopinavir, by the way, is also, as the Navir suggests, a protease inhibitor, but actually it's being used here to inhibit the degradation of ritonavir. It's a very potent inhibitor of the CYP450 system. And the idea is that again, if you are not able to make the viral RNA to incorporate new virions similar to if you're not able to make a functional virion, it's kind of okay if some of the cells got infected, but you're going to limit the infectivity, and you're not going to overwhelm the host immune response, or you're not going to elicit the massive cytokine storm. The only side effects that are really described from ritonavir and lopinavir are GI side effects, and exceedingly rare, elevations in transaminases, but those were less than 1%. And but there was no regimen that showed any change in mortality at all. So for the remainder of the kind of targeted portion, I want to talk about what I've called spike fragment vaccines. And this is an area that's exciting and I think worth some discussion on a few fronts. First, it's one of the areas of most promise for providing a definitive solution to SARS-CoV-2 or to the current pandemic. It's also interesting because vaccines don't necessarily come up that much in toxicology, but it's important to be familiar with them, especially because they're making that, because at least the Moderna vaccine is nearly entirely new. And so we may be called on to give an opinion on the toxicity thereof. So on the top is a schematic from a Nature article by Siranowski, just demonstrating what are kind of the canonical way that we think about viruses, which is on the left-hand side, you take a virus, traditionally adenovirus, and you put in the genome for, or the genetic sequence for some segment of interest, and most efforts here are focused on the spike gene. There were efforts to make vaccines against other components or other proteins in SARS-CoV-2, in particular the membrane protein, and those, none of them made them into animal trials. So you encode the spike genetic material into an adenovirus. The adenovirus goes into the cell, and it then makes a very weakened form of the virus that your immune system can recognize, can generate antibodies to, but the immune system doesn't get overwhelmed. And you can do this by, you can imagine that the adenovirus can contain the genetic material for the spike protein. It can maybe contain genetic material for adenovirus 2, depending on how large an inoculum you think is needed to elicit a sufficient neutralizing antibody response from the host. In the lower fragment, I've put a table, and this is from online post I wrote for a site, Brief19, and it summarizes three of the vaccine trials kind of are currently underway. They all target the spike protein. They're currently all in phase 3 trial, and they're enrolling large amounts of participants. The main difference between the AstraZeneca and Johnson & Johnson vaccines is should be in a logistic thing of the Johnson & Johnson one doesn't have to be frozen. The main difference between the Moderna vaccine and the other two is that it just contains RNA, and would be the first example, if successful, of a successful RNA vaccine, or of an RNA vaccine. On the next page, this is kind of the part B of the schematic, also from the same Nature paper. And I just want to take a few seconds to go through what we mean when we say a nucleic acid vaccine. So in contrast to the more canonical ones where we take the immunogenic portion of a virus and put it into a vector, here we take the gene or the genetic material, be it DNA or RNA, and we directly introduce it into your cell, or into a cell. And then if it's mRNA, it's in the cytoplasm, and it the host cell just takes it, treats it like any other mRNA, makes viral proteins, and those viral proteins then are exocytosed, picked up by the immune system, and they make a response to it. The nice thing about this is that all there is the spike protein. There's nothing else. There's no way that this can be a fully functional coronavirus. There's no way it can even be a fully functional adenovirus. Adenovirus is quite safe, as many of the clinicians on the call know, but there still are sometimes bad outcomes or severe illnesses from it. But this is just the genetic material for the spike protein. And RNA vaccines have one additional thing, which is over DNA vaccines, which is they cannot be incorporated into the host genome, so an even lower risk of mutagenesis. As the blurb in the lower right hand there suggests, the theory behind this is very compelling. They're also logistically a lot easier to produce than the full-on vaccines and the viral vectors discussed on the previous page. But there is currently no licensed vaccine for anything that uses this technology. So there's a bunch of different vaccines, and we might think that vaccines are far out of the realm of the toxicologist. But here I've put one thing that I think may seem cryptic at first. What do llamas, chimpanzees, and adenovirus all have in common? And the answer is that they were different systems used to insert the genetic material. which is to say, I went back and I said that there's two ways that you can make a vaccine broadly. You can have the direct genetic material injected, or you can use a viral vector. Well, you have to raise that viral vector in something, and that could be in a cell line of adenoviruses. It could be in chimpanzee, or it could be in llamas. And they have their benefits. And this may remind people of the differences between the antibodies against venoms that are raised in, horses, or ovine or horses, and how they're kind of slightly different because the system in which you raise the antibodies has an effect on the actual physical shape of the antibody, the amount of antibodies made. There's a question there, which thanks for that question, Elizabeth. I do want to get to that at the end because that'll take a while. So before going on to the alternative therapies, to summarize, the idea there is that we should be cognizant not only that the antibody or that the vaccine is targeted against SARS-CoV-2 or against a component of SARS-CoV-2, but we should also be cognizant of the methods of manufacture and what it's stored in, what system it was raised in, that may explain hypersensitivity reactions that are present in one vaccine but not another. The story of the COVID pandemic to date is not only one of like biotechnological innovation or drug repurposing. Early on, many people suggested and still continue to suggest alternative therapies. I thought it was useful to touch on a few, not necessarily to say how much they don't work, because they don't work, but this is a nice outreach opportunity or education opportunity, because some of the thoughts that lead people to these therapies are actually not unreasonable at all. And I think they provide a useful starting point if you're trying to build a kind of productive therapeutic alliance with somebody. So the ones that I'm going to talk about are bleach, colloidal silver, oleander, and this one, and you please, somebody who's much better at Mandarin than I am can correct my pronunciation, but I believe it's like a Qingfei Paidu. These are all therapies that have been promoted, and there may be preprints on them, or there may be articles, even fully peer-reviewed articles suggesting their efficacy. I think all of them stem from an idea of cleansing the body. Actually, the first two stem from an idea of cleansing the body and a lack of understanding of the difference between cleansing a non-living surface versus cleansing a living thing. And the other two are thought to, it's not really clear where the rationale for the oleander one, other than it might have antiviral activity in vitro. And the last one is a traditional Chinese medicine under the idea of calming the inflammation of the body. So bleach, when we say bleach, it's really, they mean miracle mineral solution, which is sodium chlorite, not hypochlorite. It's not new in the sense that it's been proposed by QAnon and by this kind of thing called Genesis 2 Church for, everything. It was most recently kind of tragically proposed as a way to cure autism, and there are some unfortunate, just very, very sad videos of people giving their autistic kids this solution, which is, more bleach-like than bleach. It's a caustic injury, and I don't think we need to go into the toxicity of it too much. The idea is that this stems from an idea that I want to kill the virus, but a lack of an understanding of this will also kind of kill me. Below here, by the way, is the cease and desist order or that the FDA sent to one of the largest promoters of this Genesis 2 church in Florida. The next one is colloidal silver, and similar to bleach, it's something that is been brought up as kind of good for what ails you, and this is something that a naturopathic doctor suggested, and then was picked up by the blogs similar to this one. It's called Geek Mamas. The actual picture of the website, which you can see in the upper left, is the My Doctor Suggests. And again, this is a naturopath doctor, just kind of all these products, if you can't see the picture, they all say silver in some way, silver lozenge, silver probiotic, silver solution, a lot of silver. And colloidal silver is, I mean, it's either goes through you, or if you absorb any of it, you'll just develop argyrism and all the attendant heavy metal poisons. What is nice and what is kind of a success story for both of these is that the FDA went after the source in addition to the general media storm and told them to stop, and they actually stopped. Oleander was reported in a preprint to have antiviral activity, and here's the preprint, and below it is a link to the brief 19 page again, where I discuss this in a bit more detail. Toxicities, your cardiac glycoside toxicity. I think the point that this touches on is that we, in the rush to find something, there's been a large amount of publications that were put out on a preprint archive, bioarchive. And you have to treat clinical data that are not peer-reviewed as interesting, but again, very, very preliminary, not necessarily something to base any treatment off of. And then it also is instructive to go back and see if this paper was subsequently published. And to my knowledge, this preprint never actually made it through peer review. And the last one is one that may be less familiar because it's out of a publication in China, and they tried this traditional Chinese medication, which I'm not going to try and say again, and they noted that actually in a kind of somewhat well-designed study, a retrospective study of 63 patients, it had no change in mortality. It did decrease one inflammatory marker. I've listed on the left-hand side, this is just a table from their paper, the components of this decoction. And there's a lot here. Some of them, the Ma Huang and the glycyrrhiza, may be familiar to the medical toxicologists among us. The idea is that there might be something here. I think that in the face of the overwhelming morbidity and mortality, we should go with kind of more proven routes. But it's interesting to note that not only do these things exist, and maybe for the research they may teach us, they may have some medicinal value, but also that some people are thinking about these things, and that it can provide a useful starting point for a conversation, because people, for example, taking this medication will at least believe that the excessive inflammation is a source of mortality from COVID-19. And I think that's a useful starting point. In 2 minutes, I want to touch on two medication interactions, and Dr. Kazzi in the comments linked to the COVID treatment guidelines. The URL is also, the first few slides. In addition to the treatment, they also have a section where they talk about medication interactions. Whether or not to use ACE inhibitors or ibuprofen was a lot of discussion early in March. The theory behind stopping ACE inhibitors is that you don't, is essentially that coronavirus already inhibits these and leads to acute lung injury. So why are we going to further inhibit them? The takeaway from that, and the same for ibuprofen, is if you're supposed to be taking the medication, keep taking your prescribed medication. Otherwise, starting or stopping it doesn't really do anything. And this is true for ACE inhibitors, for ibuprofen. It's also true, although I'm not showing it here, for statins. That's discussed in more detail on the COVID-19 treatment guidelines page. So I want to close with an extremely quick presidential case study just to tie everything together. What we see here is that President Trump went to Walter Reed after a few days after he tested positive for coronavirus, and he received three things: dexamethasone, remdesivir, and cocktail of 2 monoclonal antibodies. As we've discussed, Dexamethasone is known to help intubated patients prevent from further deteriorating. It decreases their mortality. It doesn't really work for anything else. The Remdesivir 5-day course, now he got it at like quadruple strength. That it does show some symptomatic improvement, but it doesn't change the mortality. So you might feel really good. That's not necessarily going to change the course of the battle, though. And then he got something that we don't have that much data on. There's just a small publication out recently last week with like 300 people for the viral vaccine against the S protein. And these were neutralizing antibodies, 2 different strains. In theory, though, that's great, but we have no data, really, except that one small industry-sponsored trial. And you will notice that there was no hydroxychloroquine or convalescent plasma given. So, you know, in some, we have data on 2 out of the 3 medications he got. It's Remdesivir is the one that's most clearly indicated to at least show symptomatic improvement. But for the patient population that we understand President Trump to fit in, it's not clear that any of them would change mortality. So I think now we'll move to the questions and answer. This is my contact information, mic9189@med.cornell.edu, my Twitter handle @MACACHARYA, Macacharya. And this is the PubMed ID for one review I wrote that describes some of these things in more detail. I'd like to thank all the co-authors on that article. I'd also like to thank, in addition to the ACMT and Paul Wax and Ziad, I'd also like to thank the leadership at Weill Cornell, in particular Rahul Sharma Ramarao, for giving me all the time to focus on this. And so with that, I guess I'll turn it over to Ziad.
Thank you so much, Dr. Chary. This was a great description of a dynamic process or situation that we are going through. I am going through some of the questions that we have in the Q&A, but before I do that, I would like to just mention that the American College of Medical Toxicology is actually going to be engaged in a project related to therapeutics. I'd like to have maybe Dr. Wax give a very brief overview, because I know we will present this in more details in future webinars. Dr. Wax, can you comment on the FDA ACMT collaboration, please?
Sure. Yes. And again, thank you for a great presentation. Well, ever since the early days of the COVID-19 pandemic, we were concerned about potential toxicities to therapeutics, particularly when they initially pushed hydroxychloroquine, which we knew was quite toxic in many cases. And so we've developed a program which is funded by the FDA to put on a proactive surveillance system looking for potential adverse drug events or adverse drug reactions related to COVID-19 therapeutics. And we just got funded, we just received the funding award just in the last several weeks, and we're setting up a program at 12 medical institutions across the country, essentially one of them in each of the federal regions. And we're going to deploy a team at each one of these institutions that will help to proactively look for these ADEs and ADRs, and then report them into a special data collection system that ACMT operates, and then this will be quickly passed over to the FDA for their analysis. So we're just starting it up. Hopefully it will go live over the next several weeks, and we're not sure what we're going to find, but we thought, particularly given the fact that this pandemic is going to be with us for quite a while, that we want to stay on top of the potential problems associated with therapeutics and make sure that they're reported expeditiously.
Agreed. I think having these multiple therapies available for us is great, but the healthcare providers should be alert for any potential adverse events, and remember to report these to the existing surveillance systems, such as the MedWatch, which is a voluntary reporting interface, online interface that's very useful, available through the FDA, as well as if you are part of this project, you could report it to our project as well. Now, Dr. Chary, I have a couple of questions that really talk about vaccines more than just your medication therapeutics. A couple of our viewers were asking about the AstraZeneca and the Johnson & Johnson vaccines that, as you know, are currently discontinued or paused in the U.S. and one of them in Johnson & Johnson in U.S. and Europe. Can you comment on that and maybe a little bit about the differences in these two vaccines and why, for example, the AstraZeneca vaccine requires 2 doses instead? It requires only 1 dose instead of the 2 doses required by the other similar vaccines.
Yeah. So lots to unpack there. I would say first, the pausing for the vaccine is part of kind of the usual development of a vaccine, where when there's they have a definition of some serious adverse events, and then the Data Safety and Monitoring Board will go and review the case. And remember that this already has happened once before to the AstraZeneca vaccine, and then they got underway relatively soon after. So I would not, the first thing is I would not take the pausing of either vaccine as indicative of something, some critical flaw. They are enrolling very, very large amounts of people, and the utility of that is that they can identify these rare adverse effects. And 1 out of 60,000 gets usually, it's below your 1 out of 10,000 thing. Now, as far as the different inoculum of the two, why one requires 2 doses and one requires 1, the straightforward answer is that preclinical studies showed that your titer didn't stick around. Why you need 2 of one versus 1 of the other ultimately still, at least I don't fully understand the difference between the duration of the activity. And I will say that the duration of the neutralizing antibody response is something that also we would expect to vary across population, and if somebody's on concomitant immunosuppressants. So it is something that is, I would pay, I don't think those two things are that informative. The dosing regimen can change a lot, and pausing is kind of par for the course for any sort of clinical trial.
I agree. We will have actually on October 28th a special webinar on the vaccines that will be provided by Dr. Evan Anderson. He was the principal investigator on the messenger RNA vaccine at this here at Emory University. In our pre-webinar discussion with him and with the other colleagues in this area, I'll give you a little bit of a teaser before the webinar. The issues of the dosing and the booster, the need for the booster really is dependent on the neutralizing antibodies. Also remember that the neutralizing antibodies are not the entire picture. There's also a T-cell response that is involved. And all of these things are still being evaluated. And are not necessarily predictable prior to the phase 2 trials, and now the phase 3 trials that are ongoing. The AstraZeneca vaccine, I think, is now resumed in Europe in the trial. It is still paused in the US because of the current investigation by the Food and Drug Administration. The Johnson Johnson, the AstraZeneca vaccine had the transverse myelitis, report, and that's being investigated. And the Johnson & Johnson, we don't know exactly what the adverse event was, or if it was actually in the placebo or control arm of the study. The other question I have for you, Dr. Chary, is related to steroids. I mean, I know you talked about dexamethasone. Some of our viewers are wondering about other steroids like mineralocorticoids. Also the mechanism. I know you talked about the mechanism of, restoring the inhibitory tone on autoimmune or inflammation. What about the effect on the cytokine storm or this hyper inflammatory response that is seen in children or even in COVID-19 patients, and most recently in patients with the MISA, the multi-inflammatory
MISC, I think, right?
syndrome in adult?
Oh, MISA, yeah. So excellent question. I do. There's one other that I do want to answer first, if that's okay, and that's from this Elizabeth Bilden, and it's just because I feel bad that she asked it quite early on, and I said I'd get back to it. I think I have time for both. She asked if the gene encoding the spike protein mutates, why target the vaccine there? And I think that's an excellent point. First, they tried other targets, and the only one that really stuck, so to speak, is the spike protein. So we kind of have to focus efforts there. The second is a lot of the nature of the mutations. And we don't want to get too deep into the virology here, but some of the nature of the mutations is to make the spike protein stick or dock more effectively with ACE2. And so we might view mutations as making it more difficult to make a vaccine, but that's not necessarily true. If the antibody is targeting a part of the protein that tends not to mutate, then you're kind of fine if another part of it's mutating. And so I think that's a very important concept, because going forward, a question is, well, we put all this into making a vaccine, fine, but we have to get a flu vaccine every year. So are we going to have to do this with coronavirus? And the flu vaccine is not always very effective, or I don't mean that. I mean, like, the flu vaccine is perceived as to have far less than 100% efficacy. So are we going to have little mini COVID pandemics every year? And there are some differences between the structure of influenza viruses, Orthomyxoviridae in general, and coronaviruses that mean that the coronavirus is less likely to mutate in that same way. But I think it's important that mutations it's not the case that the vaccine is like, oh, there's one nucleotide that's mutated, all vaccines are worthless. That is not the case. And then to answer the steroid question, so to my knowledge, I don't think that there's been a real in humans comparison of different glucocorticoids and mineralocorticoids. I don't think anybody has lined up prednisone, aldosterone, and dexamethasone together. But Ziad, what was the question? I'm trying to look for it in the chat. Was it?
I think that, well, the question is really good. What you just mentioned, the comparative efficacy of various steroids and the role for the cytokine storm.
So the idea is that, what's come to the forefront is the idea of this IL-6 signaling pathway. And IL-6, interleukin-6, is one of these interleukins that floats around, and it will bind to its receptor on a cell, and it has this kind of nice intricate dance it does, where it binds to an IL-6 receptor, then it binds to GP130, and then this trimer dimerizes, and this sets off a whole inflammatory cascade. And the idea is that if you were to make antibodies against IL-6, and that's where tocilizumab comes into mind, and sarilumab, if you were to somehow decrease IL-6 signaling, then you might prevent this cytokine storm, IL-6 being a cytokine. But there's two chemicals that already are pretty profound inhibitors of IL-6, azithromycin and glucocorticoids. So that's one thing. And then they also were like, well, hey, dexamethasone or glucocorticoids also rescue through this ACE2 thing. The problem is I don't think any of us are shocked to find out that steroids have multiple mechanisms of action. And then the other thing I think that it's worth mentioning with all this is we hear people hospitalized with COVID and they're given a treatment. But for some time, both in Boston and in New York, due to resource constraints and stuff, the testing for other pathogens was not necessarily as wide as it could have been. And you don't know if some of the efficacy that we see of steroids here reflects a concomitant bacterial pneumonia or something, which I think is important to say. But all these, as I mentioned or tried to point out in the Dex slide, is there's theories as to how it works. There's in vivo and in vitro data that shows that this is how dexamethasone rescues acute lung injury in rabbits. But you don't know that it's doing the same thing in humans.
Thank you. Now, I know we're getting up to 4 p.m. Eastern. Maybe we can get a couple more questions. One of them is, maybe summarizing the ideal synergistic combination of all the treatment described in the presentation.
Mm-hmm.
We can take that one real quickly, Dr. Chary, before I add a couple more questions and conclude.
Yeah, and I think that is an excellent summarizing question, and I think that something like Trump's regimen, if proven, works very well. I think that a steroid in combination with either a protease inhibitor or a transcriptase inhibitor would be good alongside a general vaccination strategy.
Thank you. And then can you discuss or maybe touch on the reinfection issue and the strategy that need to be employed to minimize morbidity and mortality from reinfection?
I wish I could intelligently comment on that. The data on reinfection, and we certainly know that there are genome sequence differences between between the coronaviruses that came went from, and I'm simplifying here, the strains that went from China to the US versus the ones that went from China to Europe to the US. We know that their sequences are different, and so it is conceivable that you could get one and then get the other. And that there would not be that much cross immunity. But area of active research, and I will go back to the bottom line up front slide I had, which is you have to social distance and not fall into this thing that, you know, I don't want to suggest too severe an analogy, but we know that there was a lot of education during the HIV/AIDS epidemic where it's not like you caught AIDS so it can't get worse. And that there were multiple strains, and I would reason by analogy with that until we get further evidence.
Yeah, I agree. I think the issue of reinfection is on the minds of many people. At least for me, you know, as an emergency physician, you know, working with this epidemic since March, at least in the U.S., we haven't seen a lot of proven reinfections. I think there are only a few case reports of reinfection in the literature. So my hope is that if this was a major problem, we would have, you know, we would have probably seen more cases of reinfection. And I do believe that there's
the issue, and I'm sorry to cut in, but there is one issue which goes along with reinfection or kind of longer sequelae, which is the brain fog, the myocarditis, and those are issues which we're only really beginning to understand as far as, you know, what happens after the initial infection.
Sure, the long-term complications of the acute infection. But as far as I think the reinfection with a new acute COVID-19, you know, infection, I think is not, we haven't seen a lot of it reported, only kind of fewer case reports. It is theoretically possible, you know, with your antibodies that you build and your immune memory. But like I said earlier, there's also other things in play, such as the T-cell response. You can also potentially be reinfected, but not symptomatic. And not transmitting of the virus, not infectious. So I think all of these have to be further studied, especially with now the vaccines that are being developed and the interaction of that with convalescent plasma. You know, all of these have to be looked at in the future. I think it's already 4:05 p.m. Eastern. I'd like to turn it over to Dr. Wax, maybe for some closing comments. We will try to address the remaining questions online and in our fact sheets that will be published in the next few weeks online as well. And Dr. Wax, would you like to make some comments and conclude?
Sure. Yeah, if we could go to the next slide, please. Yeah, again, I'd like to thank Dr. Chary for really an excellent, comprehensive presentation on a moving target, i.e. the therapeutic approach to COVID-19. And I think as of you know, September 14th or October 14th, 2020, you brought us well up to date, which is greatly appreciated. This webinar has been taped and will be available on our website sometime on Friday in 2 days, and registrants will get an email about the link at that time. We'll also try to get to some of your questions that we haven't had a chance to get to during the presentation. And again, any questions can be directed to us at the webinars at acmt.net. Next slide. As Dr. Kazzi mentioned, we'll be continuing our webinar series, which going forward will occur approximately every other week with some changes. We've been pretty much every week since March, but we'll move into an every other week mode. So on October 28th, 2 weeks from today, we'll have, as described, an update on COVID-19 vaccine clinical trials. Are we there yet? And Dr. Anderson from from Emory addressed us several months ago, and he'll provide a very important update during that presentation in 2 weeks. We'll also continue our update from the front lines segment as well, and have a talk from a physician on the front lines in Ethiopia. Next slide. Finally, all of our webinars are available online. We recently did our website, and they should be quite user-friendly in terms of accessing these webinars. You can go either to the website directly, or you can download our prospectus, which has the complete program since March, as well as upcoming webinars. So again, thank you to our speaker and Dr. Kazzi for assisting with the moderation. Everyone, please have a good day.